Today in physiology, we have started talking about the heart and different contractions that take place across the structure, some of the main external anatomy. Also, we have gone over the pacemaker cells and the contractile cells, some of the molecular physiology of the contractions and stuff like that. So we're just going to dive into it very quickly. As you guys all know, that the heart is basically divided into two pumps, the right and left. The right is basically the pulmonary and left is the systematic. And so what happens is blood or deoxygenated blood comes from the systematic body structure and drop into the right atrium, then they go to the right ventricle. Then from the right ventricle, the oxygenated blood is taken into the lungs where it becomes oxygenated. From the lungs comes back to the left atrium, to the left ventricle, and then from the left ventricle through the aorta, taken to the rest of the body to supply the tissues with oxygen. Where mainly the heart is located is it is located in the mediastinum between the two lungs and above the diaphragm. The apex is located towards the left of the body, pointing down towards the left hip. And so it is about the size of the fist. It's 230 to 350 grams. What else? I mean, that's basically the most important things that we've mentioned. Yes, so the heart is covered by the pericardium. What it does is basically a pouch that helps prevent the heart from getting a lot of friction from the surrounding structures. So what the pericardium is basically made up of the fibrous pericardium, the most external part of it, and then the serous pericardium, which is made up of the parietal and the visceral. And between the parietal and the visceral, there is the pericardium cavity that is filled with pericardium fluid that helps give the heart the space to move a little. And then this fibrous pericardium prevents the heart from overfilling. So it just keeps that steady volume of the heart. And so how the heart is situated in the pericardium is just looking at this structure right here. So basically you have a balloon. Imagine you have a balloon and you put your fist directly into this balloon. And so the air is away from your hand. You're not touching the air. However, you're just fisting that balloon, okay? Yes, so so the walls of the ventricles are mainly much more dense and stronger than the walls of the atrium. And so the thickest of all of the walls is the left ventricle, because it would push blood directly to the systematic part, which has a lot of resistance compared to the pulmonary circuit, which takes blood to the lungs and is made up of the right ventricle. That is the main power towards it. So we just need less muscle mass on the right ventricle. And let's see here. Systematic circle takes a long pathway. Yes, so that's it. There you go. So yes, that's how the fibers of the heart are arranged. So you have the bundles are crossed here, and so this crossing of bundles would make it more efficient for the ventricles to contract on the oblique direction, and so blood is more efficiently able to be pumped out of ventricles because of how these fibers are situated. Okay, so the structure of the heart in general and the valves that are involved. So the valves that separate the atriums from the ventricles are called the atrioventricular valves, and the valves that go towards the pulmonary or the aortic valves are the semilunar valves. the atrioventricular valves are the here the bicuspid and the tricuspid. The bicuspid is also called the mitral valve. So they open and close in response of difference in blood pressure on the two sides. So this means that the semilunar valves would close due to the difference in pressure between either the pulmonary circuit and the aorta and the ventricles. Here, the atrioventricular valves would close or open depending on the difference in pressure between the atriums and the ventricles. Chorda tendineae, they're attached to the valves and attached to papillary muscles that are protruding from the ventricular walls. And what these do is basically help close those atrioventricular valves when they're needed to be closed. Okay. So how blood moves, or how the duction of this takes place, is that blood would come to the right atrium from the superior vena cava and the inferior vena cava and drop into the right atrium, and then directly flow into the right ventricle. And so then the contraction of the right atrium takes place to get rid of the extra blood left in the right atrium. Then after that, blood would move from the right atrium into the pulmonary circuit to the lungs and go to lungs. And here on the opposite end, blood would come from the lungs into the left atrium by the four pulmonary veins, and then so the right atrium would pump blood into the left atrium would pump blood into the left ventricle. And so you can see here a large mass of muscle would contract to push blood into the aorta and then lead it towards the body. So the valve openings and closings. So what happens is when the ventricles are relaxed, the semilunar valves in both of those are closed because pressure in the ventricles are lower than the pressure in the aorta and the pulmonary circuit. So we need those closed to prevent movement of blood back into the ventricles from the systematic and the pulmonary circulation. And then so you have the other valves, the atrioventricular valves. These valves are closed when the ventricles contract to push blood into the systematic or the pulmonary circuit. We don't want blood to move into the atriums because it already came from there. So we want a unidirectional movement of blood. Yes, so I just explained that. Okay, so the cardiac muscle is striated. It has a sliding filament mechanism. They're short, branched, and interconnected. Yes, they have pale, centrally located nuclei. The interstitial spaces filled with connective tissue have many capillaries. And it has a fibrous cardiac skeleton, which acts both as tendon and as an insertion, giving the cardiac cells something to pull on, exert their force against. Also, the skeleton prevents the spread or irresponsible spread of excitability or electricity from the atriums to the ventricles, because this has to go through a very structured pathway from the sinoatrial node to the atrioventricular node, to the bundle of His, and then up the Purkinje fibers. And so what happens is those cardiac muscles are or have a lot of gap junctions and a lot of desmosomes. And the desmosomes, they keep the myocytes from pulling apart. So when it contracts, it holds the whole structure of the muscle. That's the function of desmosomes. And the gap junction between the cardiac fibers help in allow those myocytes to be electrically connected. So allows all the excitements to be spread regularly. Yes, so as we said, desmosomes, gap junctions. What else did they say here? Yes, there's a lot of mitochondria for, of course, for the production of ATP, because contraction here is needed, and so energy is needed. T tubules are wider and fewer than in skeletal muscle. This is because we don't need a large number of T tubules, because T tubules are not the only supply of calcium. Hence, the other way those muscles will take in calcium is from the blood circulation. And so we don't have cisterns like in skeletal muscles. So triads here do not exist. The cardiac sarcoplasmic reticulum is simpler and lacks the large terminal cisterns, as I said here. Okay, so this is the molecular pathway in which contraction and relaxation of the muscle of the heart occurs. So what happens is that instead of action potentials arriving and causing change to this calcium channel, what happens is, well, yes, so action potential comes in, the change happens to the calcium channel, and instead of this calcium channel removing the RYR, what happens is calcium would go in from the extracellular matrix to this receptor, causes change into it, and then that would cause this channel to open up, releasing calcium from the sarcoplasmic reticulum into the cell inner environment, and then that will cause changes to the actin myosin filament, by the interaction with troponins component C, that causes change into the tropomyosin and release of actin active sites. So then would allow myosins to attach to the actin and so cause contraction to be possible. Now, when we want to relax the muscles, two things happen, is that the active transport of calcium ions would be from the inner environment of the cell into the sarcoplasmic reticulum, and also transport or active transport of calcium outside the cell into the extracellular matrix. These two things help reduce calcium concentrations inside the cell. The second pathway here, we're talking about 0.9 and 10. So what happens is ATP is used to exchange, what is it called? Yes, so the energy of Na plus taken in would drive calcium outside. Now, to keep the balance of the ions, Na plus K plus ATP pump would work, so that causes K plus to move in and Na plus to move out. So this is basically the molecular function or functionality of the contractile cells. So this is just him drawing all of those things. If you want it, you can pause. Yes, and so this is called calcium-induced calcium release. Calcium-induced calcium release, okay? That's the relaxation. Now, so what happens is cardiac pacemaker cells. We're going to explain basically how they contract and how they relax. So here, this is called the pacemaker potential or pre-potential. How this pacemaker potential rises. So this is caused by intake of Na plus, and so when it reached into the potential, so this is just a normal increase in action potential because Na plus is the active ion that allows for action potentials. Now, when the threshold is reached, calcium 2 plus is taken in from the environment and causes a change to cause the release of calcium from the sarcoplasmic reticulum. This increases until it reaches this point. Now calcium channels close, K plus channels opens to cause repolarization. And then IF channels open. I forgot the function of the IF channels. Just give me a second. So IF channels there. Yes, IF channels open due to hyperpolarization. Polarization. 1 second. Yes, so yes, to return. Yeah, now I remembered. Where do you go? Yes, to return the potential. So we're basically, what we're doing is we're at a very low point in hyperpolarization. So to return the potential to the threshold, we open IF channels to allow Na plus again, right? So to reach the threshold, and then Ca2 plus takes over. Okay, so this is the motion of the electrical impulses through the heart. So the sinoatrial node is the main heart pacemaker, causes impulses, has a frequency of 75 impulses per minute. And so if this has a fault in it or it doesn't function regularly, other parts of this pathway takes over. The atrioventricular node has a delay of 0.1 seconds. This is very useful to allow for all of the blood to move into the ventricles before the ventricles contract, okay? The atrioventricular bundle or bundle of His. So here, SA node, AV node, bundle of His, then go up to Purkinje fibers. Okay, this is the pathway of the electrical pulses. This allows for a very good contraction. So contraction by the atrial node allows the atriums to contract. Atrioventricular node awaits so that the ventricles are filled. Now contractions go down, up, back to the base, allows for the full contraction of the ventricles to cause blood to move to the systematic and the pulmonary circulation. And so pacemaker potential and the contractile cells potential, pacemaker potentials are a bit different. And so this is the contractile cell. Why there's a plateau is because, I forgot, 1 second. Yes, this gives enough time for the heart, enough time for the next contraction to take place, gives time for the muscles to relax before its second contraction. And so Na plus channels would open to cause this spike. Na plus channels will close. Then the plateau is at phase number 2. Calcium channels open and fast K plus channels close. So calcium would be going in and K plus is not allowed to go out. So this causes the plateau. Now the repolarization happens when calcium channel close. So calcium is not allowed to go in. Slow K plus channels open, so allows for very slow outburst or outflow or efflux of potassium outside of the contractile cells of the heart. And then so finally the resting potential. This is the contractile process. Okay, so yes, so here, there we go. Why this phase or this process takes place. This long plateau in cardiac arrest has two consequences: contraction is sustained, so that blood is ejected efficiently from the heart, and there is a long refractory period, so the titanic contractions cannot occur and the heart can fill again for the next beat. So titanic contractions do not occur in the heart, and contraction is sustained, so that blood is ejected efficiently from the heart. So yes, so okay, these are the reasons for this type of contraction. Okay, so stages. Late diastole. So what happens is blood arrives and blood is flowing directly to the ventricles. Okay. And so number two, atrial systole. This here, we're pushing whatever is left in the atriums to the ventricles. That's the atrial contractions. The isovolumic ventricle contractions. Here we're having that all of the valves are closed, because now slowly the pressure in the ventricles are building up. So what happens is atrioventricular valves are closed, and the semilunar valves are not open because the pressure in the systematic and the pulmonary circulation is still higher than the ones in the ventricles. Now the ventricles pressure increases above the systematic circulation and the pulmonary circulation. The semilunar valves open, okay, and so ventricular ejections happen, so blood moves into those things. Now isovolumic ventricular relaxation, what happens is the pressure, all of the valves are closed. This is because the pressure in the what is it called? The pressure in the atrium, okay, is still less than the pressure in the ventricles, okay, and the pressure in the ventricle is still less than the pressure in the systematic circulation. And so atriums right now are relaxed to take in the blood that has come in from the pulmonary and the systematic circulation. And then we go back again to late diastole. Okay, and so how we're going to explain this by graph. So we have the pressure and we have the volume. And so in the pressure graph, we have it divided into colors. And so it also shows you what stage the heart is at. So here we're collecting at this stage, the first one there, just before this point right here, the number one there, the heart is collecting blood from the systematic circulation and the pulmonary circulation going directly into the blood, into the ventricles without anything. Now, atrial systolic, atriums contract, right, to push out whatever blood is left in it into the ventricles. Now, at the EDV here, 2A, blood in the ventricles is slowly building up. The isovolumic contraction, where all valves are closed, right? So slowly building up until it reaches this point. Once it reaches this point, now the blood is moving. So the blood is moving and wants to go out wants to go out through, so wants to move out. So the pressure within the ventricles is way higher than the pressure within the aorta. This opens the valves and so allows for the blood to completely move into those circulatory systems. Now we're starting to decrease, right? So why? This is because now the aortic valve is getting closed. That's the dichrotic notch. It's closing because now we don't want blood to move back from these aortic or the pulmonary circulations into the ventricles, because the ventricles just contracted and starting to relax. So this is the drop right here. Okay, and then And then we have a decrease in the blood pressure. See here, the rise in the aortic pressure is because now the aorta is receiving all of this blood. Now it's receiving all this pressure. Now the increase in this pressure directly happens. And so decrease in number 3, we're having the atriums now are starting to receive the blood. They're giving it in a relaxed manner to the ventricles, and then up until here where we are. Yes, so yes, the left ventricle. The left ventricles are relaxing. So when it's relaxing, it will just take some time until all of the blood is taken in, until we have another contractions from the atriums to push whatever blood is left. Okay, so let's explain it here using the volume. So now when the ventricles are getting filled up, right, we're gaining more volume in the ventricles because of more blood moving in. Now contractions happens from the atriums, hence volume increases a bit. Now blood pressure starts to, the pressure within the ventricle starts to increase. Once the ventricles contract, now volume of the ventricle starts to decrease, that it is pushing blood into the systematic and the pulmonary circulations. And once everything moves into the aorta, starts to relax again to receive more blood, hence an increase right here, and then atrium contracts to push in more blood, and so more volume happens. Okay, I hope this one was easy to understand. I hope I explained it well. You just need to get your mind around it. These are the sounds. I didn't pay attention much to it, but what I understood from here is that there are 2 sounds. The mitral valve closes before the tricuspid valve. That's voice number 1. The aortic valve closes before the pulmonary valve. That's the sound number 2. Yes, so how parasympathetic and sympathetic nervous systems affect the rate of the heart. The parasympathetic causes the action potential to have more time to be reached because it spaces this out. It decreases the rate at which we reach the threshold, where sympathetic stimulation causes less time for us to reach the threshold. If you see here, this is longer. It's decreasing the frequency. Here, it's increasing the frequency. That's sympathetic effect. You can see here. And so parasympathetic neurons, how they work is they reach to the muscarinic receptors. They increase K efflux. So K plus, more K plus is going out. This causes repolarization, right? Does not really help us. We don't have Na plus here. We have K plus. So K plus is effluxing. We're causing repolarization effects. Look here, hyperpolarizations right there, and decrease Ca2 plus influx. So this does not help us in getting anywhere. Hyperpolarization cell and decrease of depolarization, heart rate decreases. Sympathetic, on the other hand, activates B1 receptor, beta 1 receptors, increase in Na plus and Ca2 plus influx. So more of the positive ions inside, rate of depolarization increases. You can see here, rate of polarization increases, heart rate increases. That's how they function. Okay, we need to know those pathways. Yeah, I remember what he explained here. He said that, let's see, yes, if the blood receives more, if the heart receives more blood from the systematic circulation, this causes more stretch. And if there's more stretch, then there's an increased force of the heart, causing it to increase the stroke volume. So always remember that the more the heart is stretched in the initial process, the better it will be in creating more force and more blood. That's what he said. Okay. And so these are the certain things that we have to know. This is the cardiac output formula, heart rate times stroke volume. And you have to know those definitions just in general. And I think that's it. Yeah, I didn't understand that part. Actually, I stopped paying attention here, but just for you, he also said just you have to keep those 4 things in mind. But I'll just check that later. Hope this was useful. I hope I didn't take much of your time, but I'll see you guys in the next one.